Hey everyone, and welcome. Today, we're diving deep into the single most incredible molecule in the known universe, DNA. This is it, the blueprint of life. It's the code that determines pretty much everything about us, and we're going to unpack the amazing story of how we found it, how it works, and what it all means.
So here's our game plan. We're going to start with a century-old mystery that had scientists stumped. Then we'll get up close with the famous DNA double helix. After that, we'll see how this master blueprint gets copied perfectly, how its code gets turned into actual proteins that do all the work in our bodies, and finally, we'll talk about the insane project to read our entire human instruction manual. Let's get into it.
All right, so our story kicks off with a really fundamental question. For ages, people knew that kids inherit traits from their parents, right? You get your mom's eyes or your dad's nose. But for the longest time, nobody had a clue how that information was physically passed down. What was this mystery substance?
Now, back in the 1860s, you had the brilliant Gregor Mendel on his pea plants. He worked out all these amazing rules of inheritance and called the things being passed down factors. But that's all they were, abstract ideas. He couldn't see them or touch them. So the biggest question in biology became, what are these factors actually made of? That one question drove research for nearly 100 years. The first real clue popped up just a few years after Mendel in 1869. A guy named Friedrich Miescher found this weird acidic stuff in the nuclei of cells and called it nuclein, but nobody really connected it to genetics for decades. The big aha moment didn't come until 1953. That's when Watson and Crick published their famous model of DNA. And it's so important to remember, they couldn't have done it without the critical work of others, especially the stunning X-ray images from Rosalind Franklin and Maurice Wilkins. Her data basically revealed the helical shape, and that was the key that unlocked the whole puzzle.
So, what was this world-changing discovery? Well, Watson and Crick revealed a structure that was just so beautiful, so elegant, and so perfect for its job that it basically changed everything we thought we knew about biology. So let's take a closer look at the architecture of life itself.
And there it is, the double helix. It's one of the most famous shapes in all of science, right? It's basically a twisted ladder. And believe it or not, this simple shape is the secret to everything DNA has to do. It's not just pretty, it is an absolute masterpiece of design, built perfectly to store tons of information, and crucially, to be copied over and over again with incredible accuracy.
Okay, let's break this ladder down. The two long sides, the backbone, are made of repeating sugar and phosphate molecules, making them really strong and stable. The two strands are what we call anti-parallel, which is just a fancy way of saying they run in opposite directions, kind of like a highway. Now, the rungs of the ladder are made from four chemicals, or bases, A, T, C, and G. And this is the magic part. There's a strict rule. Adenine, or A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. A with T, G with C. That's it. This rule is the absolute foundation of genetics. Now, DNA is the star of the show, but it has a super important sidekick called RNA. Here's a good way to think about it. DNA is the master blueprint locked away safe in the cell's nucleus for long-term storage. But to actually build something, you need a working copy you can take to the construction site. That's RNA. It's a messenger. They're very similar, but RNA has a few key differences. It uses a slightly different sugar, it swaps out the base T for a base called uracil, or U, and it's usually just a single strand. These little changes make it the perfect mobile messenger molecule. Okay, I need you to pause for a second and just think about this. If you took the DNA from a single one of your cells, just one, and stretched it all out, it would be over 2 meters long, about 6 feet, and all of that has to fit inside a nucleus that's so small you need a powerful microscope to even see it. I mean, how on earth is that possible? It's an insane packaging problem.
Well, nature came up with a genius solution. The cell uses these little proteins called histones that act like tiny spools for thread. The long DNA strand wraps itself tightly around these histone spools. This forms a structure that looks kind of like beads on a string, and we call those beads nucleosomes. Then, that string of beads is coiled up and coiled again and again until it's packed down into this incredibly dense structure called chromatin. It's a truly brilliant piece of biological engineering. So we have this amazing blueprint, and it's packaged perfectly. But every time one of your cells divides to make a new one, you need a perfect copy of that entire blueprint. So that brings us to the absolutely vital process of DNA replication.
You have to love this line. This is from Watson and Crick's original paper, and it has to be one of the biggest understatements in the history of science. They're basically saying,
Oh, by the way, the AT and GC pairing we just figured out, yeah, it kind of tells you exactly how this thing copies itself. The structure itself held the secret to its own replication. Just amazing.
And here's how it works. The process is called semi-conservative. Imagine you unzip that DNA ladder right down the middle, separating the rungs. Now you have two single strands. Each one of those original strands then acts as a perfect template. Because of that strict ATGC rule, the cell's machinery knows exactly which new base to add. The result? Two brand new identical DNA molecules, where each one is made of one old strand and one new strand. It's just an incredibly elegant and foolproof system.
And get this, it happens unbelievably fast. The main enzyme that does the building, DNA polymerase, can add about 2,000 of these base pairs every single second inside a bacterium like E. coli. Think about that. It's a tiny molecular machine working at lightning speed with almost perfect accuracy. It's just mind-boggling.
Okay, so making a perfect copy of the blueprint is one thing. But how does the cell actually read the instructions on that blueprint to, build stuff? This is where we get to the really cool part, going from a static code to a dynamic protein. So the flow of information in a cell follows a really clear path, and we call it the central dogma. First, as we saw, you have replication, where DNA makes more DNA. The next step is called transcription. This is where a small section of the DNA, a gene, is copied into that messenger molecule, mRNA. Then, that mRNA message leaves the nucleus and goes to the cell's protein-building factories for the final step, translation. This is where the message is read and the protein is built. So just remember that flow, DNA to RNA to protein. That's the core of it all.
Now, to go from the language of DNA, which has only 4 letters, A, T, C, and G, to the language of proteins, which uses 20 different amino acids, you need a dictionary. You need a way to translate. And that's exactly what the genetic code is. It's the universal set of rules that the cell uses to read the DNA language and translate it into the protein language.
And this code has some really neat features. First, it's a triplet code. That means the cell reads the bases in groups of 3, and each 3-letter word is called a codon. Second, it's unambiguous, which means a codon like, say, AUG always codes for the same amino acid, always. But it's also what we call degenerate or redundant. This means that several different codons can code for the same amino acid. This is actually a great feature because it acts like a built-in buffer against typos or mutations. And maybe the most amazing thing of all, the code is pretty much universal. The same codons mean the same thing in you, in a mushroom, in a fly, in a bacterium. It's a shared language that connects almost all life on Earth.
So a gene is basically a sentence in the DNA book. It's a specific stretch of DNA that has the instructions for building one specific protein. Now, in organisms like us, our genes are a little weird. They're often split into pieces. The parts that actually contain the coding information are called exons, but in between them are these non-coding chunks called introns. So before the message can be read, the cell has to do some editing. It snips out all the intron bits and stitches the exons together to make the final, readable message.
All this incredible knowledge about how genes work and how the code is read, it naturally led to one of the most ambitious scientific projects ever attempted. The mission to read our own instruction book, the entire human genome from beginning to end.
And the scale of that challenge, it's just hard to comprehend. The complete set of our genetic instructions, our genome, is made up of roughly 3.3 billion of those chemical letters. Not million, billion. The goal was to figure out the exact sequence of every single one of those A's, T's, C's, and G's.
So back in 1990, the Human Genome Project kicked off, and its goals were huge. It wasn't just about reading the sequence. The plan was to identify every single human gene, to make all of that data totally public and free for any scientist to use, and to build better tools for analyzing it all. And really importantly, they also set aside money to study the massive ethical, legal, and social implications, the ELSI, that would come with this powerful new knowledge.
And in the end, that's what we got, a complete reference map for our species. For the first time in history, we had our own instruction book. This has completely transformed medicine, allowing us to find the genetic roots of diseases, and it's also given us a much, much deeper understanding of our own evolution and how we're connected to the rest of life on this planet.
So that's where we'll leave it. We've gone from a time when we didn't even know what a gene was made of to a point where we hold the complete sequence in our hands. It has been an absolutely incredible scientific journey. And it leaves us with one final, very big question. Now that we can read the entire blueprint of life, what do we do next? What should the next chapter of our story be?